Hello friends, in this video I will explain about the replication of stringent plasmid. Replication of stringent stringent plasmids. Okay, and we know that there are two kinds of plasmids. One is the uh, known as the relax plasmids, and another one is the relax, and another one is the stringent. Okay, relaxed plasmids are those which maintain a high copy number actually in the bacterial cells or bacterial cells you can say, and stringent plasmids are those which maintain very low copy number 1, 2, 3 are known as stringent plasmids. Okay, in this video we are particularly discuss about the replication process of stringent plasmids, how it initiates. Okay, so in case of stringent plasmids of the PSC 101, it is a kind of you can say a stringent plasmids, and it's maintain copy number about 1 to 3. And the origin replication, for replication there is for example, if you consider this is the site, and here there is an important protein which is actually necessary for this replication, and this is called rip A protein. Rip A, this is very important protein for replication in plasmid, stringent plasmids, and it actually bind within a region which is near the original replication of PSC 101. Okay, for example, this is the origin of let me erase first these things. For example, if you see that this is the site for origin of replication, then here after some base pairs, after some base pair there are actually sites where which is known as iterons. For example, this is one iteron. There are actually 3 to 7 iteron sequences, 3 to 7 iteron sequences, and each of them actually 21 or 17 to 21 you can say length, base pair lengths there. For example, here 3 iteron sequences are present, and after that there is a site where the this is the gene site for the repair protein, and is synthesized and bind with this sequence. Okay, repair protein binds with this iteron sequence. This sequence, this is the repair protein repair, and this one, these sequences are actually iteron. iteron, these are called iteron. Okay, let me erase these things. This is the original replication site, this is the iteron sequences, this is the repair protein here, it's synthesized and binds with the iteron sequences. This is the repair site, and here is this one is the site for the DNA binding protein, that is the DNA A. We know that DNA A, DNA B, those things are also required for the origin of replication. Once it's the repair protein binds with the iteron sequences, there is another protein known as integrated ISC, integrating host complex. It actually binds in the origin of replication site of the plasmids, and this region is actually AT rich, and as a result what happens is that the sequence become DNA become bends, and this one is the AT rich region. This region is AT rich, and as a binding of ISC it become bends, and here now the DNA A binding site, and the iteron sequences now are close to each other. This is the iteron sequence, and with the help of the repair protein they now actually bind with each other and start the replication process from here. And this is actually theta shape replication actually starts from here. This is the origin of replication procedures. And regarding the regulation process, once the copy number is high enough, the replication process need to stop. Now what is they do is that now the repair protein, once the plasmid's copy number is high, now they actually bind with another region within their near the they actually bind with their promoter. There are two inverted terminal repeat sequence are also present just besides the iterons. For example, these yellow colors are the iteron sequences, and these two are inverted terminal repeats, IR1 and IR2. These two, and they bind with these sequences, and they actually down regulate their own production when the plasmids copy number is high enough. Okay, high enough, it down regulates their own production. And there is another process also when the plasmids copy number high. For example, this is one plasmid and this is another plasmid, this is another plasmid in the cell. So as a result of their high copy number, what they now do is that they actually joining two plasmid with each other with the help of their repair protein. For example, this is the iteron binding site of this plasmid, this is the iteron binding site of this plasmid. Now the repair protein actually joining two plasmids each other, as a result the plasmids now unable to initiate their replication process. They, as a result the plasmids copy number become decreases, and this procedure of joining two plasmids with each other is known as handcuffing. CUFFTING. So this much you should know about the stringent plasmid replication. I hope you like this video. Thank you. If you like, please share with your friends. Thank you.